All right, welcome everyone. In this particular lecture, we're going to be talking about the biological testing of biomaterials. And I'm pretty excited for this particular chapter. I think it's one of the more interesting ones in the series. In the past few lectures, we've talked about the biological background of biocompatibility and what can go wrong. And in this chapter, we're learning how to apply that so that we can actually test for the biocompatibility of a particular device. So on that note, let's review first what the definition of biocompatibility is, and it's the ability of a material to perform with an appropriate host response in a specific application. Now, on one hand, it's an appropriate definition, and I honestly don't think there's a better way to define it. However, it's incredibly vague. It doesn't really tell you much about anything. And for the most part, it really boils down to just there's a lack of anything bad happening. And as we see throughout this chapter, really a definition of good biocompatibility is simply a lack of anything bad happening. So to provide a little bit better introduction to biocompatibility, I'll talk about four main factors that perhaps could make something bioincompatible. The first one being the toxicology of a particular device. Now, it seems obvious that a biomedical engineer would use non-toxic materials for a particular implant or biomaterial device, but that's actually not the concern of either the engineer or the FDA. What is a concern is some of the leachable extracts that remain within a polymer or metal, usually left over from the fabrication or storage process, that could potentially cause problems. These might be unreacted monomers for a polymer implant, catalysts or initiating fragments, stabilizers, process additives, or any metal and glass ions or fragments left over from the bulk of a material. There's also reactions from extrinsic organisms. These would be materials left over after a sterilization procedure, so I don't want this to be confused with live organisms due to poor sterile handling, which might cause an infection. These are fragments left over after a pathogen has already been killed, but could still initiate an immune response. There are also mechanical effects that could be caused by localized irritation or rubbing, abrasion, or moving of an implant against localized tissue. Even mechanical mismatches where the bulk properties of the device is different from that of the tissue, for instance. Or even sharp corners as opposed to round corners. So biomedical engineers, they hate sharp corners on implants. They avoid them at all costs and want nice rounded corners. Lastly, this was the focus of last week's lecture about the cell biomaterial and tissue biomaterial interactions. We talked about the concept, one of my favorite concepts, of frustrated phagocytes, where they actually try and ingest an implant much bigger than themselves and they're unable to. So they sort of figuratively cry out cytokines and inflammatory mediators, and any cytokine releases that can initiate the inflammatory process. Now, one actually straightforward, although admittingly invasive way to measure biocompatibility is to look at the foreign body capsule that forms around a biomaterial. Now, foreign body capsule is really the result of normal inflammatory responses and the foreign body reaction that's preceded by granulation tissue formation in the wound repair process. And it's really a collagenous monolayer, which is a scar tissue, separating a biomaterial from the regular tissue. And it's sort of a way of isolating a biomaterial and forming this capsule around it to isolate it from the rest of the body. It happens to all foreign bodies. So again, just because that there's a capsule around a biomaterial doesn't mean it has poor or good biocompatibility in itself, but we can look at it and sort of infer the level of biocompatibility. A quote-unquote good biocompatibility characteristic would have a really thin capsule. It's only 2 to 3 layers, avascular, and really dense layer around that biomaterial. Poor compatibility, on the other hand, would be really thick, extravascular, so you can actually see these differences in pictures A and B, one being really thin. I see few vascular structures, it looks dense, and they all contrast to what I see in picture B. This is on the same scale, so of course it's much thicker. It's looser. I'm starting to see some formation of angiogenesis and vascular structures. And then as we move on to number C, which has no real orientation, I see a lot of disorganized cell tissue growing, multitude of inflammatory cells. That's what all the sort of dots and granular-like patterns are. And if we were even able to see the foreign body giant cells, which are characteristic of a really poor compatibility reaction, then we could conclude that that device was not compatible. Now, the schematic that we have before us helps describe the process that occurs to establish the preclinical biological safety of a biomaterial. The first step involves a chemical characterization of all the materials used in a particular device, including both the bulk materials and then also the trace leachables that are present and left over that I described previously. Now, besides just describing the quote-unquote ingredients of a particular device, we also want to do material and surface analyses so that we could perhaps understand the route of exposure a little bit better, which goes into the next assessment, which is the toxicological risk of those chemicals that were just previously characterized. What's involved in this is first identifying what hazards each chemical in the previous step present in a particular device, then understanding what kind of dose or exposure that particular patient would get from the device. Then you would determine the chemical's dose-response relationship to understand how those hazards correlate to a particular dose, and then finally combining all of those to characterize the exact risk to a patient. Now, very often, only those steps are sufficient in order to progress a device to clinical trials, meaning testing in humans. This particularly occurs whenever a device is already based on an established predicate and only minor changes to the device are being made. For example, a change in the supplier for the particular polymer that's being used to make this device. If certain flags are present, or perhaps the risk has triggered a certain unknown in that particular material, these tests might progress towards the in vitro testing, which might look at the cytotoxicity, genotoxicity, and carcinogenicity testing, which we're going to talk about in the next portion of the lecture. And even, again, at that point, that still might be sufficient to pass and progress the device towards clinical trials. Or, again, if they raise any concerns, that might progress towards in vivo testing, which tests many of the same exact things that we just tested in the in vitro testing stage. Again, at this point, this might pass and the material might progress towards clinical trials. Or if enough concerns are raised through in vitro and in vivo testing, the engineers might have to go back and change the materials that they're using. This in itself will be called a trigger event, which requires the need to change the materials being used and undergo more testing. Certainly other things can cause this as well. Like I said, a change in the vendor for a particular material, a supplier change, the use of a different material as a substitute in an already established device, or maybe adverse events associated within the clinical trials themselves. Now, when previously discussing the toxicological risk assessment, I mentioned the need to create a dose-response relationship for a particular chemical used in a biomaterial. Some of the factors that affect this quote-unquote dose are the route or site of exposure, the duration that biomaterial is left into contact and thus can leach out its extractable components, and then also the frequency in which you might apply a particular biomaterial. So whether it's a long-term implant that's meant to stay in for 10 years, or a catheter that's meant to perhaps repeatedly being exposed to a patient every day. Those things can all change the frequency and thus the dose that particular material might be exposed to a patient. A dose-response relationship is thus created by plotting some quantifiable response or adverse event along with any number of doses that you want to test. Now, there's three things commonly reported from these dose-response relationship. The first is the no observable adverse effect level, which is the highest concentration that was tested where no significant response was detected. The next one being the lowest observable adverse effect level, and as the name implies, it's the lowest concentration that was tested where a significant adverse response was observed. So the lowest is above the no observable adverse effect level. And then we have the margin of safety or exposure, which is the ratio of the no observable adverse effect level to the quote-unquote worst case dose received. So the largest material, perhaps for the largest patient with the most surface area, whatever device configuration can give the possible highest dose for that particular device. In this case, the bigger the margin of safety, the lower the safety risks. So we want a large number for that. Now, one thing I like to concentrate on are methods. I want us to make sure that we have a practical understanding of how people actually test for biocompatibility, as opposed to just talking about this at a conceptual level. So I'm going to go through a number of tests that describe some of the aspects I just mentioned before, like cytotoxicity and genotoxicity, and I want to show you pictures on what the data looks like and how these tests are actually carried out on the benchtop or in the lab. So the first one in the in vitro testing stage is cytotoxicity, which is really just the quality of being toxic to cells. This can happen by a number of different ways. One is by altering some of the local biological environments that cause a general molecular or cell dysfunction, or a chemical can act specifically with a particular endogenous target molecule, a ligand that somehow disrupts cellular function directly and specifically, causing the cell to die. Now, in general, to prep a sample for cytotoxicity testing, and many of the tests that actually we're going to talk about in this chapter, involve a few steps and have a few requirements associated with them. Any chemical prep step should not cause any significant chemical changes to the extract that you're testing. You want to use both polar and nonpolar compounds so you can extract all types of chemicals and leachables. And then also you want to conduct them at appropriate durations and temperatures, like 37 degrees Celsius, that mimic the actual device implantation or use. Now, a very common cytotoxicities test is called the MTT assay. It's used to directly measure cellular metabolism and then, by that, indirectly, cell viability. The way it works is that it makes use of an endogenous cellular enzyme that reduces a water-soluble MTT dye into an insoluble purple form. Now, because this enzyme has to make use of the cell's NADPH cycle in order to work, only wells with viable cells in the presence of these leachable extracts are going to produce this purple color. So that way we can measure the darkness or how purple these solutions are in order to determine how many cells you have alive or how many cells you have dead. Next up, we have testing for genotoxicity and carcinogenicity, which all fall under the umbrella of mutagen testing. Genotoxicity is defined by the potential to induce an undesirable effect on DNA or any of the genetic process within host cell or tissue. This could be DNA destruction, gene mutation, or chromosomal aberrations. Carcinogenicity, on the other hand, is the ability of a substance to cause cancer. Cancer itself can be caused through DNA damage, so a carcinogen can also be a genotoxin. It could also disrupt other cellular metabolic processes that don't fall under DNA damage. Now, a very common in vitro test that tests for mutagens is the Ames assay, which uses a salmonella strain that in itself cannot produce histidine and requires histidine to be supplemented in the media in order for that salmonella strain to survive. For this test, you would have a control that has no possible mutagen or leachable extract from your material, and then also your testing apparatus, which has the mutagen in it. You would then plate both salmonella strains in those two test conditions in separate plates with media that has no histidine in it, or minimal, and you would incubate that for some time. Now, in theory, for the control sample, because the salmonella strain is now lacking histidine, it's going to die off eventually. The reality of that is that there's actually natural forms of mutations that can occur, so there might be some viable cells left over that have gained enough mutated ability to create their own histidine. And what you want to do is you want to compare that to your actual tested material and compare the amount of mutations that occur that now allow that salmonella strain to survive on its own. Reproductive toxicity studies are a specialized type of in vitro and in vivo tests that usually aren't carried out unless there's some sort of reason or supporting studies to indicate that this would be a concern. Usually these are performed alongside cytotoxicity studies with the MTT assay, in part because we want to be able to differentiate what's toxic to cells in general and what's specifically toxic to reproductive cells. So in order to do this, you would have two treatment samples where one are non-embryonic stem cells representing general toxicity and then embryonic stem cells representing the reproductive cell toxicity studies. In addition, you would also want to monitor the differentiation of these stem cells after being incubated with your test material extract for 10 days. You would visually monitor them to determine what dose inhibits 50% of the embryonic stem cell population. This concentration is termed the ID50. In the MTT assay, you would determine the IC50, which is the concentration necessary to kill 50% of the cells. Testing for blood compatibility is going to be its own separate portion of this lecture, but I want to touch on a few in vitro tests for blood compatibility here. In general, you can measure for thrombosis visually doing some sort of macroimaging, or even down at the macro and nanoscale using scanning electron microscopes or SEMs. You can measure for the level of coagulations in general using the thrombin, antithrombin assays. Remember, antithrombin is one of the body's natural inhibitory mechanisms for coagulation. So the more of this TAT compound that you have in the body, the more inhibitory mechanism your body is forced to put out. Thus, a poor thrombin, antithrombin concentration would indicate poor blood compatibility. We can also measure the activity of platelets and how they aggregate together by labeling them with a radioisotope like indium-111 and seeing where they aggregate and concentrate together. We can use assays that measure the activation of the complement pathways to determine their immunogenicity. We can also do a few different hematology tests. The picture on the right here is a hemolysis assay, which measures the disruption of red blood cells, which gives a red coloring to the plasma solution, so a material that had poor blood compatibility that would cause red blood cell lysis would be a darker red color. But all these tests in general have a number of different disadvantages, in part because blood is so complex. So first of all, these tests require physiological relevant test conditions. And blood flow is not a static thing. It's very dynamic. It's different for different parts of the body. So it's tough to test and have a one-size-fits-all test for any biomaterial need. And because of that, there's generally a low level of clinical relevance on the in vitro side. So these almost always require in vivo studies. Next up, fibrin, thrombin, and platelet activity is highly impacted by injury and surgical trauma. So you might have good compatibility results in a controlled lab environment that are not indicative of what would happen at the time of implantation of a biomaterial. And finally, it's hard to replicate the full test conditions of especially large devices because the surface area and surface chemistry are so highly impact blood compatibility. You can't just do this with a simple leachable extract test. It's not the same. Now, at the beginning of the lecture, when we talked about the safety evaluation schematic, I mentioned that neither in vitro tests nor in vivo tests are necessarily required for the preclinical stage. You can sometimes go right to the clinical setting if your device requires it. Certainly, there's practical reasons for not having to do in vivo testing, but also ethical reasons. Unless there's something that indicates in the in vitro stage that you have to use it, these tests aren't done. So unless you have a newly developed biomaterial or something in the vitro stage is indicated otherwise, you don't do in vivo testing for just general fact-finding tests. You might, however, do it on the final product form after you have the final manufacturing and sterilization condition completed so that you can minimize the amount of tests that you have to do. Now, there are a lot of different types of in vivo tests. I'm only going to cover a few of them. They can range from sensitization and irritation to full reproductive toxicity, biodegradation tests of your implant in long-term in vivo studies. You can look at the complex immune responses that you couldn't otherwise see on an in vitro scale because cells don't have immune systems. Hosts or animals have immune systems. So let's talk about a few of them. Sensitization tests are used for a lot of biomaterials and also for a lot of cosmetic tests. They generally test for allergies and intolerances to a particular material and their extracts. Guinea pigs and rabbits are commonly used. That's why if you look on the back of a shampoo or a moisture label, you can see the crossed out bunny sign, which indicates that there was no animal testing. You can also do general intramuscular or IV injections of chemical extracts, just like in the in vitro tests. You want to replicate and extract leachable components in the implantable state. So you use the final form of an implant. You would mimic its surface area and the right thickness of a particular material that you want to test. And then you can go on to a host of different types of toxicities that test toxicity at a number of different scales and local or systemic effects. So for instance, systemic toxicity tests the toxicity of material not at a localized site, but at systemic levels at organs away from the point of contact. Here you might measure the pyrogenicity of that particular material, which is its ability to induce fever. And then also the localized toxicity at a number of different time levels, from the acute stage, which is within 24 hours, all the way up to subchronic and chronic toxicity that last on the scale of the lifetime or percentages of the lifespan of a test animal. Genotoxicity studies in general look for the same abnormalities that would be indicated in an Ames assay. The most common kind of genotoxicity test is the rodent micronuclear test, which tests for chromosomal damage. Here you would test visually what kind of chromosomal or nuclear damage you would see. So for instance, in picture A, you see what's called a budding nucleus. That is starting to cleave off of the main nucleus. You'll get a micronucleus, as in B and C, which is a tiny little nuclei fragment off in the cytoplasm of the cell. Or you could have what's called a notched nucleus, which doesn't have that nice spherical shape, but actually has a notch in it, just like here in picture D. Implantation tests are probably the most intuitive type of biocompatibility study because they actually involve the implantation of the targeted implant device. This would be a great example where you look for the foreign body reaction to a particular device. So you'd be thinking of taking histology samples of the tissue surrounding a device after extraction. And I should also note here that this is not a study for efficacy. So you're not actually testing the function of a particular device, just the host reaction to it. So you can actually put the device wherever is most convenient for the study and not necessarily at the exact site where it's ultimately intended. And then finally, I'm just going to cover the carcinogenicity tests. These require, as you can imagine, long time implantation of a device in a particular animal because cancer takes a long time to develop. So because of the long timeline required, these are very often done in conjunction or simultaneously with chronic toxicity studies. Now, as you can imagine, both because of the time required and the costs associated with housing and caring for animals, these are rare and only performed if previous data indicates a need for this. Now, one unique aspect of the carcinogenicity test that we have to consider is what's called the Oppenheimer effect, whereas the tendency for non-biodegradable biomaterials to induce spontaneously the growth of tumors in rats particularly. So whenever we're testing devices in rats for carcinogenicity, we always have controls not of just simply a lack of a biomaterial, but a substitute material of comparable form and shape, usually made out of some known substance like polyethylene, so that we can have a better control. Because we know that the polyethylene is going to induce some amount of tumor growth, and we want to see a relative comparison to the intended device and its tendency to cause cancer. So now we're getting into blood material interactions, and in part because blood is so tricky and complex, I want to focus on this one a little bit more in detail than I did the rest of the chapter. So going back to our definition of biocompatibility, we have the ability of a material to perform with an appropriate host response in a specific application. We can have a similar definition for blood compatibility. Now, if you're looking for something more definitive and specific than biocompatibility, I'm afraid this is not what you're going to get. Because the definition that Buddy Ratner gives here is the property of a material or device that permits it to function in contact with the blood without inducing an adverse reaction. So once again, in short, biocompatibility or blood compatibility is the lack of anything bad going on. Not very helpful, buddy. Thanks. First, let's discuss some common biomaterials or biomedical devices that have vascular use. There's the heart-lung machine used during surgical procedures, hollow fiber hemodialysers used during kidney failure, catheters which just give general access to blood. There's heart assist devices which assist the heart. There's of course stents, heart valves, and synthetic grafts that are all used during surgery or even just during the life of the patient. Now, in order to establish the need for a good blood-compatible device, I want to highlight there are a few instances where there are no current devices available that have good compatibility, like the artificial heart. People that have artificial hearts now simply use them as a bridge until a heart can get donated to them because it's not going to last the lifetime of a patient. There's also no such thing as a long-term oxygenator for an artificial lung. There are no intravascular biosensors, and there's also no vascular grafts smaller than 5 millimeters. To highlight that even more, in general, many devices are only considered blood compatible and safe when they're used with anticoagulants or antiplatelets. And these are not just during the wound healing process, but during the lifetime of the device. There are a number of reasons why these devices could fail. Heart-lung machines have a tendency to produce severe bleeding and microemboli. Heart valves and catheters can shed emboli that can lead to stroke as that blood clot travels to the brain and occludes blood from getting where it needs to be. And also synthetic grafts can lead to ischemia and even downstream tissue damage when they're deprived of oxygen. Because of this, devices that come into direct contact with blood nearly always require regulatory testing. Now, going back to the definition of blood compatibility is the lack of adverse reactions associated with blood. Now, in general, really what we're talking about here are the blood material interactions, or the BMI assessments, which in general just look for the thrombogenicity of a material. Thrombogenicity is the extent in which a device or material, when employed, induces an adverse response, some of which include a few of these examples, like a disruption in blood flow in artificial grafts. They can hinder the mechanical motion of heart valves. Embolisms can form downstream of the actual device within blood vessels. They can hinder gas exchange through oxygenators. They can cause the chronic destruction of certain blood components and hemolysis. They can also produce mediators that can continuously induce inflammatory reactions through complement or other immune responses. Now, any of these blood material interactions can occur on the surface of devices, within blood solutes. They can interact with blood proteins or even cells like red or white blood cells. Example of this could be different absorptions of proteins, absorptions, so the diffusion of these proteins within the actual device, the denaturization of proteins, or activation of platelets. Now, all these factors depend on 4 different items, 3 of which are classically described in VITRO's triad, which are components of the blood itself, the surface of the device, and then the flow conditions. So again, not just being a static condition, but high shear rates, low shear rates, or oscillatory flow. The 4th component, which we're going to add to this, is also time. So the time in which the device is in place of something or in contact with blood. Now, because of the complexity of the device that you might want to test, and certainly also the complexity of blood and the conditions in which blood interacts with the devices, there are a number of difficulties and complications that can arise in attempting for test for BMIs. One is that we can't extrapolate results obtained under one set of test conditions for another that has any of those 4 parameters changed. Okay, we also can use short-term testing to predict long-term results. In general, we can't predict in vivo device performance based on blood material interactions in idealized in vitro flow geometries. Also, many types of blood contacting devices and even the device design itself is going to interact BMIs of the materials used. Also, manufacturers of various devices are reluctant to discuss specific chemical compositions in which they tested at, so it's even hard to get data on blood material interactions associated with different types of materials or even vendors. There's also few comparisons between human and animal blood. These are so species specific that observations in one animal's BMI is going to have a wildly different result than we might have in humans themselves. Also, it can be very expensive to measure thrombogenicity in a systemic way. So locally it might be one thing, but to measure it on a systemic level requires an entire animal and only one type of device being used in that animal at the time. So incredibly complex. To highlight some of the difficulties, particularly on the last point where we have either a localized versus systemic model that we're trying to look at, I want you to imagine a few different possible testing scenarios and outcomes of a single test. So here we have our tested device. It looks like a shunt, for instance, that's exposed to blood, and we have 4 different outcomes. The first one being the ideal result is that in the middle column, we have the actual event that happened during exposure, which is nothing. And then whenever we took out that material, we see no clot form. That's great. Scenario B highlights what we would expect to see with a poorly blood-compatible device, where we actually get a clot, as we see in the middle column. And then whenever we extract that material, we see that that clot is there. We conclude it's not compatible. However, imagine a case where we have a thrombus that embolizes and then goes downstream from the device, like we see in scenario C. So what actually happens is that thrombus forms, it sheds, and that emboli then goes somewhere downstream. So whenever we extract the device and we see what's ever in the last column, we see nothing's there. So we might conclude that it's actually compatible, no thrombus formed. That sort of complication could also be seen in scenario D, where what actually happened was, yes, a thrombus formed and it broke up. This is still indicative of poorly compatible device, but yet we don't have evidence because that thrombus isn't there whenever we extract it. However, there is a reason why we do these tests, so hope is not lost. Okay, so for instance, both in vivo and in vitro testing has been very helpful in defining mechanisms for blood material interactions. So whether or not we can correlate, that's a difficult and a separate part, but certainly they can outline mechanisms for blood compatibility. Now, for instance, both in vitro tests and small animal species are very useful to screen for big differences between materials. Also, large animals are useful for identifying significantly reactive materials. Now, because blood almost coagulates instantly when encountering foreign substances, all in vitro and in vivo tests typically require anticoagulants in order to successfully test for their BMIs, which you might think throws off the conditions of these tests. But remember, most biomaterials that get implanted almost require constitutive anticoagulant therapy. So we can still make correlations and decisions when we have anticoagulants. So even though we can't infer decisions in cases where we wouldn't have anticoagulants, that's not always the case, because devices routinely require anticoagulant therapy. Now, on the topic of anticoagulants, I wanted to revisit this diagram on coagulation so I can highlight what some of the synthetic anticoagulants do. So one of the major ones that we have are known as vitamin K antagonists. Vitamin K is a necessary enzyme that produces prothrombin, the precursor to thrombin, a major activator of platelets. So for instance, warfarin is a very common vitamin K antagonist and a heavy-hitting anticoagulant. There's also antithrombin 3 activators. So antithrombin binds with thrombin to prevent it from activating platelets. Heparin is something that makes antithrombin 3 or fourfold more efficacious. The factor X cascade is something that I very briefly talked about. It's part of the necessary pathway that converts prothrombin into thrombin. And then there's also calcium chelators. Calcium is required in a number of these cases. It's factored heavily in the intrinsic and extrinsic factor X cascade. So again, another medium for preventing the formation of thrombin from prothrombin. Sodium citrate, EDTA, are very common calcium chelators that are used very often in the blood. If you likely collect blood or you donate blood, the vial in which it gets collected in is very likely treated with one of these two substances. Furthermore, you also have antiplatelets that themselves prevent platelets from activating. ADP receptor inhibitors are a common example for this. ADP is a very common activator of thrombin. And then also we have thromboxane inhibitors. Thromboxane is necessary to, again, activate platelets. And this is actually what aspirin does, is that aspirin prevents the function of thromboxane. So if you ever have a headache and you have a high amount of aspirin, I'm not sure if you've ever noticed and perhaps cut yourself, but your blood tends to not coagulate if you have large concentrations of aspirin in you. Now, because Vikar's triangle mentioned that blood flow is an important parameter for understanding BMIs, we're going to talk a little bit about shear forces on blood and how blood flow controls the rate of transport of platelets to surfaces. They can do this through one of two ways, either through diffusion or convection. In reality, it's a mixture of both, but one of these is usually a controlling factor in blood coagulation. Under low shear stress or low flow conditions, we have what's known as diffusion-limited adhesion. In this case, we want to think of almost having a limited amount of platelets at the site because they're not being brought in by a high flow. So what we're limited by is the diffusion of those platelets to the surface and not the reaction of the platelets when they're finally at the surface. Now, under these conditions, fibrin is abundant. We don't have a lot of platelets, but we do have red blood cells, so we form what's called a red thrombus. It's composed mainly of those red blood cells inside a fibrin mesh. The opposite condition, which occurs under high shear stress or high flow, is what's called reaction-controlled adhesion. In this case, platelets are numerous because they're being brought in at a really high rate, and we're only limited by the actual attachment or reaction rate at the surface of the device. Now, in this case, we form what's called a white thrombus because it's formed mainly of platelets, less fibrin, and not as many blood cells. Now, there are a few examples of different in vitro tests that we can give. I'm going to briefly cover some of those and then go into a little bit more detail in the in vivo tests. Methods in general can involve any number of very simplistic things. They could involve placing blood in a container made of the test material. They could involve placing the test material in a container or vial with blood. Or they could involve something a little bit more complex like circulating blood through physiological flow conditions in contact with that material. One very simple example of the latter test I just mentioned is the Chandler loop. These Chandler tests involve a loop of your particular material that has blood within it, and then that simply gets rotated through a water bath at the right flow conditions at 37 degrees, so that eventually a clot will form within that tube. Again, we can't really use these for optimization, so we can't determine small differences between materials, but we can screen for materials and potentially eliminate ones that are highly reactive towards blood. Some of the things that we will look for are parameters such as the whole blood clotting time, the activated partial thromboblastin time, or the whole blood recalcification time, in which we use blood that's treated with a certain calcium chelator, like I mentioned in the anticoagulants part before, and then we recalcify it to see at what point it finally coagulates. Now, before moving on to some of the in vivo BMI evaluation tests, I'll first cover a few concerns and reiterate some of the limitations of these type of tests. So for instance, timing or the type of measurement may exclude important blood responses. This was highlighted just about 5 slides ago when I talked about the 4 different outcomes of a particular extraction test for a model material. Of course, blood is species specific, so the responses in animals can differ from that of humans. And also, we can't control blood flow conditions. So, certainly, other animals have different blood flow rates compared to humans, so that's hard to control in itself. And also, the trauma from the implantation of the material, of course, can cause local thrombus formation coagulation in itself. So, it can be difficult to infer what is a naturally forming thrombus due to the blood material interactions and what's due to the surgery itself. Those are some of the generalized limitations. I'll talk about one of the more common in vivo BMI evaluation methods, which is called chronic shunting. This is where a shunt is made that implants in one of the arteries of an animal and then recirculates back into a vein. What's nice about this method is that you can actually control the blood flow somewhat easily, and you can even measure it through visual means itself. You can use either native or anticoagulated blood, depending on your test setup. You can also take advantage of natural physiological coagulation controls that the animals would have itself, so antithrombin and the like. Also, this can be used to test both short and long-term BMI evaluations. Now, because blood has many different components, there's a lot of different tests that we can use, and there's even a variety of assessment methods for each individual component. For instance, thrombus, we can visually assess the formation after an explant study. We can also use radioisotope accumulation, like with indium-111, and see where it aggregates. This can also be done with platelets through radiolabeling again. We also have platelets counting through visual assays. Red blood cells hemolysis is a very common test done for BMI evaluations. We can also actually count them visually to determine the decrease in cell count. You can do the same thing with white cells as well, and other enzyme assays that are specific to different leukocytes. There's also evaluations of different coagulation factors like fibrinogen. I mentioned the II.3 assay a number of times. Again, that complex of antithrombin 3 with thrombin can give a great indication on a material's BMI. And then finally, we can look at activation of complement through different complement protein assays. And all these can look at the different consumption rates, the dysfunction, or even activation and increased presence of any of these components to give us a variety of answers for blood BMI evaluations. And there'd be a number of reasons why you would only look at certain components or only do certain assessments depending on what kind of device or what kind of times they're interacting with blood. So for instance, devices that have small surfaces and interact with blood only on short time scales, such as catheters or guide wires, your concern might just be significant thrombus formation because any sort of clot is going to absolutely hinder their use. Even if you get a small clot, that's going to include a catheter. On the other hand, you might have large surfaces that only interact for short times, like dialysis units or pumps and oxygenators. Here you look at the depletion of blood cells and proteins, perhaps immune and inflammatory responses, or systemic organ dysfunction. And then there's also devices of any kind of size that would be implanted for long times, like heart assist devices, where you look at full systemic effects and organ dysfunction. I'll now end with describing some of the more sophisticated ways in which we evaluate different components of blood. The first one being light scattering. Now, in general, light scattering is just used to determine the size of particles in suspension. So in this case, we might use it to determine the size of aggregated platelets or thrombus formation and the size of that thrombus. What light scattering does is that first it emits a laser pulse of light at particular particles. Whenever that light hits the particles, it's going to scatter, and that amount of scattering is detected within the light scattering unit. And then subsequent pulses that also cause scattering are used to infer the relative movement. So for instance, a very small particle that moves fast through a media is not going to scatter light nearly as much in the same way as, say, a very large particle that's not moving a lot, that's going to have very much the same type of scattering as you had from the preceding lights emitted. Another very sophisticated method is called flow cytometry. Now, the first step in understanding flow cytometry is that you first have to label whatever it is you want to detect, whether it's a cell like a platelet or other red blood cells, white blood cells, certain proteins. You can do this very easily with specific antibodies that themselves might have a fluorescent tag, and you can even incorporate multiple different fluorescent tags to detect multiple things simultaneously. Next, this mixture of cells and proteins is flowed through the flow cytometry unit, where a laser that shines on top of the fluorescent tags and emits a light is detected and quantifies the level of that particular fluorophore. From there, you can even actually get sophisticated units that also incorporate light scattering units that can measure the size of the cells, and then can actually also sort depending on the location of those fluorescent tags, or maybe electrostatic deflectors that divert different droplets into separate containers. So you can actually separate out cells in your proteins into different collection vials. Now, in conclusion, what I really would have liked to have ended this lecture with is a list of good blood compatible components that are commonly used today in different conditions. Unfortunately, there is no list of just great materials. There's some materials that are used in some conditions, like for low shear blood flow conditions, we like to use heparinized surfaces, hydrogels, or perhaps non-fouling materials like those with self-assembled monolayers or that use pegylated materials. For high shear blood flow conditions, we like to use silicones, Teflons, or any hydrophobic polymers made up of N-alkyl chains. However, none of these materials compare to what's considered the most compatible material, which is that of the living epithelial monolayer lining of blood vessels. Not only are they an idealized surface, but they also have natural inhibitory mechanisms that fight off coagulation and even prevent it in the first place. So in conclusion, just kind of take away from this that thrombosis occurs dynamically and continuously, always undergoes both formation and dissolution. So device failure is not just one that causes coagulation, but is one that results in a weak imbalance of these processes. So our body has its own natural inhibitory mechanism. So even if a device causes thrombosis, we can fight that off. And that actually happens even in the perfect conditions all the time as well. Now, your choice of material is going to be dictated by a number of different things. It could be the nature of the flow of the actual device or the blood across the device, the mechanical properties required by that device, certainly the length of time that they're going to be in contact with the blood, maybe even the condition of the blood as well, whether or not you're going to have anticoagulants in the blood or not. And there's also to consider the actual consequence of an embolism from forming. There's some conditions where that might be less risky than others, so certainly that opens up some possibility, whereas others, it just narrows your choices. So hopefully now from previous lectures, we have a better understanding of what can go wrong and some of the biological and cellular mechanisms that dictate that. And now we know a number of different ways from which we can test it, why it's important, where some devices have failed, and why in other cases it might be more acceptable to have less compatible components than in others. So thanks for listening.